Good morning. My name is Raghav Sehgal, and I work as a postgraduate researcher in Dr. Richard Kibi's lab at Yale School of Medicine. I'm also part of the IOMIC facility at Yale, which is a core facility that specializes in understanding metabolism by performing untargeted metabolomics, fluxomics, and oxygen consumption experiments. For our talk today, we will be discussing one of the projects which is ongoing at the Kibi lab and IOMIC facility. The project is Drug Metabolism Database, or as we call it, DMDB. Now, before we talk about what DMDB is, I would briefly like to explain why we felt the need for a tool like DMDB. The first and foremost reason is our limited understanding of how a lot of FDA-approved drugs work. To give some numbers, for 18% or 1 in 5 drugs approved by the FDA, there lacks a clear understanding of drugs' mechanism of action. While for 2 out of 3 drugs, we do not have a clear sense of off-target effects. This lack of knowledge translates to limited clinical effectiveness of these drugs. Thus, we feel there is a clear need of a data resource that can enable us to understand mechanism of action as well as off-target effects better. Secondly, a resource with an exhaustive list of effects of a drug can have a lot of benefits. The benefits could potentially be drug repurposing. Compounds often for one indication might have an on or off target effect that could benefit another indication. A drug database can help in identifying compounds which could be used for treating diseases like very different for which they were approved for. Another example is combination therapy. A compound which might not be effective independently because of compensatory pathways in a disease could be combined with another drug which targets the compensatory pathway. A drug database can help in identifying complementary compounds which target a specific compensatory pathway. Now, given the impact of pharmaceuticals on metabolism, a curated integrated omics study to catalog the metabolic pathway impact of different drugs in different tissues can significantly aid in helping us understand the effects of drugs better, while also allowing us to find candidates for drug repositioning. Thus, we feel an integrated omics database which uses metabolism to understand mechanism of action of drugs can have a lot of potential value. Now let's talk about DMDB 1.0. The first phase of DMDB has 2 cell lines, INS1 rat insuloma and C2C12 mouse myoblast, each treated with 10 drugs targeting a variety of cellular and metabolic processes. For each cell line and drug combination, 4 different omics data sets were generated, essentially metabolomics, transcriptomics, fluxomics, and phenomics. Data from all different cell lines and drugs is combined in one unified database called DMDB 1.0. Let's briefly talk about how different data sets were generated for this project. First, let's start off with metabolomics. For metabolomics, we used a mass spectrometer, specifically the SCIEX 6600 QTOF, in data-dependent acquisition mode. The chromatography columns used were hypercarb and C-arene columns, while the polarity modes were positive and negative. In total, for each sample, we generated 4 different data points covering the complete metabolomic spectrum using a combination of the 2 columns and the 2 modes. For metabolite annotation, we used 2 different approaches, both targeted and untargeted. For the targeted approach, we used a 600 IROA standard library curated by MSMS annotation. For the untargeted approach, we annotated 2200 features from the dataset. Targeted and untargeted results across the 4 separations were then merged, giving priority to targeted metabolites, resulting in 2700 unique features. For the purpose of transcriptomics, we used the Illumina HiSeq 4000 sequencing platform. Transcript annotation was done using deep learning models. For fluxomics, we used a uniformly carbon-labeled D-glucose and 1,2 carbon-labeled L-glutamine labeling strategies. The mass spec used was SCIEX 5500 with differential motility in MRM method. Mass isotoper multi-ordinate spectral analysis was then performed on the data to find relative fluxes for the central carbon metabolism. For the purpose of phenomics, we use Seahorse XF analyzer to generate cellular oxygen consumption data. All of the data generated in the previous steps was then processed using the AI-omic pipeline. AI-omic is an artificial intelligence-enabled integrative omics pipeline that has allowed us to swiftly process terabytes of data generated in the Drug Metabolism Database project. It uses a combination of machine learning, network optimization, natural language processing, and automation to generate swift and accurate insights. You can know more about the AI-omic pipeline on our poster with the ID WP310. Now let's briefly talk about the data visualizations offered by DMDB. DMDB has a multitude of data visualizations and analysis that it can perform. But here we are specifically looking at one of the visualizations offered by the database where you can look at KEGG maps of different drugs overlaid with different types of omics data, in this case specifically metabolomic and transcriptomic data. We will talk more about these KEGG visualizations in our forthcoming slides. Now it is useful to talk about why DMDB and AI-omic combined are such a unique platform in themselves. And there are 4 specific reasons for this. The first one being it allows for true integration of omics data. Now very often integration of omics data refers to generation of multiple omic data sets and viewing them in parallel. AI-omic on the other hand integrates all 4 of its omics data to view on the same KEGG network. Secondly, it's the speed and accuracy offered by DMDB and AI-omic. Our data generation methods combined with the AI-omic pipeline can generate insights for 10 drugs in less than a week with high accuracy. This speed and accuracy is very unique to DMDB and AI-omic. The third is automation of insights. One of the biggest challenges in large multi-omic data sets is to find succinct biological insights by manually scavenging the data. AI-omic automates this by using publicly available information to find relevant insights using natural language processing. And last of all, it's the benefit of the comprehensive view of metabolism. DMDB with its 4 omic datasets and integration gives a holistic and multi-pronged view of metabolism at different levels of the cellular process. All 4 of these reasons combined together make DMDB and AI-omic a very unique platform to be used for research. Now let's briefly talk about how this tool can be used. I wanted to show a brief video demonstrating some of the functionalities offered by DMDB. So let's have a look. What we're seeing is the default view here, which essentially is all the different KEGG maps for all the drugs and the different cell lines. Now we're going to change it to a specific KEGG visualization for puparlisib in INS1. Here we can look at the metabolomic and transcriptomic data on the KEGG map itself. We're going to talk more about this particular visualization in our forthcoming slides. Again, the tool has a wide variety of visualizations to offer. We're going to look at the pathway visualization now for puparlisib in C2C12, which integrates metabolomic, transcriptomic, fluxomic, as well as oxygen consumption rate data, all in one network. Again, we're going to look slightly more in depth for this particular visualization as well in our forthcoming slides. Again, the goal to show this video was to just give a small demonstration of the kind of visualization the tool can offer. Now let's have a quick look at puparlisib, one of the drugs that was offered by the Drug Metabolism Database. Now, puparlisib is a PI3K alpha inhibitor. Its potential use is in different types of cancer where PI3K-AKT pathway is of crucial significance. It is presently in clinical drug trials, but recent results show low efficacy with high toxicity. Now, the goal of looking at puparlisib is not to delve on how the mechanism of action of puparlisib works or how or what off-target effects it might have. It is more to show how we can really use the visualizations offered by the tool to generate insights on these particular scenarios. Now, one of the views that we recently talked about was the KEGG visualization. Now, let's dig a little deeper into it. What we're looking at here is boparlisib in C2C12 cell line, and what you can see is a KEGG network where the nodes are metabolomic data points and the edges are transcriptomic data points. The blue color represents upregulation of the drug with respect to the control, while red represents downregulation of the feature with respect to the control. The size of the node represents log fold change, while the shade represents significance. If we look at the KEGG maps of boparlisib in C2C12 and INS1 side by side, we see an interesting observation, which is boparlisib seems to have a lot more significant effects in C2C12 cell line as opposed to INS1. If we dig a little deeper, we find a few of the changes in the two cell lines are very similar. These could potentially be common on-target or off-target effects of the drug. Similarly, changes which might show up only in a specific cell line would also be very interesting, since these would be off-target effects potentially caused by the difference in tissue or organism from which the cell line was extracted. Now, last of all, I wanted to show the integrative omic map or the pathway visualization, which integrates metabolomic, transcriptomics, and fluxomics data along with oxygen consumption rate data. Here, just like the previous KEGG map, the metabolites are represented on nodes, transcripts are represented on edges, while relative flux is represented on dashed edges, as you can see, or arrowed edges. And on the right hand side, we have the oxygen consumption rate information. What's interesting here is to see that the maximal respiration and ECAR, which from oxygen consumption rate, are almost halved for boparlisib with respect to the control. And you see a similar effect in the TCA cycle as well, where most of the metabolites and transcripts and fluxes of the TCA cycle are all significantly downregulated. Now, again, this gives a very interesting insight as to how metabolomic and transcriptomic and fluxomics data is related to phenomic data and could potentially generate very interesting insights. Now, again, the goal of this was to just give insights into how this tool could be potentially used to generate new insights about drugs, the mechanism of action, and potential off-targets. Our next goal is called 555. The goal is to generate data for 500 drugs in 50 cell lines in the next 5 years. To do this, we are looking at adding new cell lines for the 10 drugs, new drugs, and new omics data such as proteomics. We imagine this tool to be like TCGA in the future, where different labs work together to understand how drugs work better and in turn create a public repository of data for the greater good of science and human health. Now, last of all, I would just like to briefly thank all the people who have contributed to this project. I want to give a huge shout out to the IOMIC core facility at Yale University and the Kibi lab, which has allowed us to do this project and make it grow. Shout out to also the Yale Center for Genome Analysis, which helped out in the transcriptomic data, SciEx, Elucidata, as well as National Institute of Health. For any questions, thoughts, or comments on DMDB, reach out to me, raghav.sehgal@yale.edu, or my PI, richard.kibi@yale.edu. And for potential collaborations with IOMIC facility at Yale, contact rebecca.cardone@yale.edu. Thank you.